Okay, welcome everybody. It's 12 noon, so I'm going to get this started right on time. My name's Stephen Balk. I'm at Beth Israel Deaconess Medical Center in Boston, and it's my great pleasure today to introduce Matt Friedman for this issue of Connect Science. Before I do that, a couple housekeeping items. I just wanted to remind all the participants that the Connect Science presentations are all available online at the Connect Science website, and you're encouraged to go back and look at them, any that you may have missed over the past many months. The second point is that Matt will take questions at the end, and I will kind of MC those questions. So if you have questions, don't use the chat icon, but use the Q&A icon to post those questions, and then in the end I'll read them off or combine them, and we can go till about a little after 1 in responding to questions, and if there's some further questions, then I'm sure Matt will be able to respond to them by email. So I'm going to do a very brief introduction. Matt went to undergraduate and medical school at University of Michigan, then internship and residency at University of Pennsylvania, and then came to Boston and the Dana Farber Harvard Cancer Center to do oncology fellowship, and he's stayed here since then and driven through the ranks and has become really a leader in the area of genetics and epigenetics. He's had a particular initial interest in GWAS, and most importantly, I think, in kind of functionalizing SNPs in GWAS, including, I think, probably the first to show that the 8q24 SNP that's associated with many cancers, including prostate cancer, actually interacts functionally with the MYC gene. He's most recently done a lot of work looking at, in prostate cancer, at the androgen receptor and epigenetic kind of modification of androgen receptor and where it binds, and some really interesting data that there's reprogramming going on that is related to developmental reprogramming that occurs during development. So hopefully I haven't given away any punchlines, and with that short and probably inadequate introduction, I'm going to turn it over to Matt. Thanks very much, Matt.
Thank you, Steve, and thanks for the opportunity to present our work. As Steve mentioned, I'm really going to talk about, I'm going to keep things quite broad, and it's really going to be sort of almost a retrospective of what our lab has been investigating and our thoughts really over the past 6 or 7 years of looking at the non-coding genome, or what was formerly referred to as junk DNA. And I'm really going to do that through the perspective of 3 different vignettes. And again, if we don't get through everything, I think that's okay. But there's going to be 3 different stories that I wanted to talk about if we have time. One is, as Steve had alluded to, it looks like the metastatic program, at least in prostate cancer, hijacks a developmental epigenomic program from fetal prostate. The second vignette is really going to be about how non-coding elements can actually be oncogenic drivers, and really has reshaped my thinking of how gene-centric we are, and how important the non-genic elements are as well. And then the last, if we have time, I'll get into some work that we're doing in liquid biopsies, where we're again using epigenetics to look at, in this particular case, to identify early stage kidney cancer. So I really, again, I'm going to keep things sort of broad and as I was putting the talk together, what are some things that have emerged over the past 6 or 7 years as we've been gaining knowledge? So I think that this is a little bit of a different data type. I think it's emerging, and I just wanted to sort of throw some thoughts out there for people to think about as I'm going through these stories. The first one is that I think that there are insights that can be gained through the systrome, and I'll use systrome and epigenome interchangeably, the terms. And that's to say that I think there are insights that are gained through the epigenome that aren't gained through looking at other data types, such as genetics or transcriptomics. Again, this second point is really just a reflection and something that we've observed, that the epigenetics tends to be a less noisy data type than transcription. The third one is there's not a single epigenome, but there's multiple epigenomes depending on what you're looking at. And one of the things that struck us are that there are some marks that tend to be very stable and there are other marks that are dynamic. The fourth point here is, I think, a really open question in the field, that there is reprogramming, but how is that reprogramming being enacted? That's something that we're actively thinking about. And then finally, how do perturbations, including therapeutics, how does that affect the epigenome or systrome? I think all of these things I don't have the answers to a lot of these, but I think that these are the types of questions that we're starting to really think about. So I'm going to spend probably 5 or 10 minutes going through an introduction just to center everybody. I hope I'm going to make the case by the end of this that the non-coding genome is not junk DNA. And I want to go back to when I was in medical school. Steve said I was at the University of Michigan. We were fortunate one of our professors teaching us medical genetics was Francis Collins. This was the textbook that we used. And I went and I dug it back up. And as you can see in the table of contents, there's really nothing about the non-coding genome, because we didn't learn about it and things were very, very protein centric. And this part of the slide is to show that this is a display that's at the Wellcome Collection in London. And there's 109 volumes that they have here that represents our human DNA sequence. And if you open up one of these volumes, this is what it looks like, a bunch of A's, C's, G's, and T's. There's 109. And what's fascinating to me is that we've spent so much time thinking about the 1.5% to 2% of the genome that codes for proteins, which is essentially 1 or 2 of these volumes out of 109. So there was this notion of just an emphasis on the protein coding genome, almost to the exclusion of the non-protein coding genome. And people did initially dismiss anything outside of proteins as junk. And our entree into this, again, as Steve alluded to, was really through genome-wide association studies. So I trained in human genetics, not epigenetics. So what am I doing in epigenetics? And the answer really lies in this slide. So each ball here is a trait that's been mapped to a particular location on the genome. And here's the karyotype of the human genome. And over the past 15 years or so, genome-wide association studies have illuminated where common variants are in association with common traits. And I'm talking about this writ large. So cancer, what are the predispositions to cancer, to inflammatory bowel disease, to cardiovascular disease, et cetera, et cetera. And the big punchline from this 15 years of work is this, that the vast majority of these variants that have mapped for GWAS that control predisposition to human traits are located outside of protein coding regions. And this is really in stark contrast to Mendelian disorders, which is what I did learn about in medical genetics, Li-Fraumeni syndrome, BRCA1 and 2, where the mutations were in protein coding regions. If we're talking about common diseases and non-Mendelian, over 90% of variants that lend themselves to disease predisposition are outside of the protein coding regions. So that was our entree into the non-coding genome. Now, we get a hint about what these variants are actually doing through a parallel effort that was going on. So there was a bunch of activity in GWAS that was going on over the past 15 years. And there was also an effort called ENCODE, which was to annotate the epigenome. was a completely distinct effort, but this is one of those happy coincidences in science where these two fields ended up informing each other. So what people started doing is they took these variants that I showed you on the prior slide and asked, are they enriched in any particular feature across the epigenome? And the answer is unambiguously yes. So what's currently thought is that many of these variants that predispose to common traits are either creating or destroying transcription factor binding sites and affecting gene regulation. And that's really been sort of, I think, one of the major hypotheses that's emerged from GWAS and epigenetics over the past, I'd say, 10 years. This was really, can people still hear? Can people still hear me?
Yes, we can hear you.
Okay, because it looks like my screen froze.
Yeah, we can hear you and see the screen, so I'm not sure.
Oh, there we go. Okay.
You better?
Yeah. Okay. So really, again, just to contextualize what I'm going to be talking about, I think that this is a useful framework in how to think about things. And this, I think, was first really advanced by Bert Vogelstein. That's often referred to as the Vogelgram, which is really the state transition. So we can go from normal epithelium to localized disease, to advanced disease, to treatment-resistant disease. And each one of those state transitions can be characterized by a particular data type. And what we've done really well is where I put the check marks for genetics and transcriptomics through things such as TCGA. Those have really informed what is changing across these state transitions. And I think in the past 5 to 7 years, tools have really emerged that now are allowing us to look at these state transitions through the perspective of epigenomics and epigenetics. And one way that I think about this is that really epigenetics is where tumor sequencing was about 10 years ago. So my screen is frozen again.
Okay, I can hear you, and you're on the epigenomics slide is what I see.
Yeah, I'm trying to advance it, but it's not advancing. Let me see if I can. I don't know if I can move this to. Oh, yeah, it's just a sort of some slow advancing. So what is epigenetics? Again, this is just to sort of center everybody who may not be as familiar with this type of work. At the end of the day, we have about 5 to 6 feet of DNA that we have to cram into a cell, each cell. And that's done through its wrapping around histones. And the way that genes are turned on and off, the way that they're regulated, are through histone modifications, as I've denoted over here. And I'll talk about this H3K27 acetylation, which you just need to think about this as a mark that annotates active promoters and enhancers. I'll talk a little bit about access to chromatin, accessible chromatin versus non-accessible chromatin. We measure that through ATAC-seq, and DNA binding proteins through transcription factors. So all of these elements come together, and the net result is really turning a gene on or off. And again, just very quickly, these are the tools that we're using: chromatin immunoprecipitation followed by high throughput sequencing. And what happens, what this technique allows you to do is to chart out where a particular protein of interest resides across the genome. And so we can get these genome-wide maps of proteins of interest. And again, I'm really going to focus on the androgen receptor here. So we're going to chart out where the androgen receptor is sitting across state transitions. Or if we want to annotate regulatory elements such as enhancers and promoters. We can use this histone mark, H3K27 acetylation, and make these maps about where regulatory elements are on across different cell types in different states. Same thing for ATAC-seq. This will let us annotate where open or closed chromatin is in a given cell type. And this slide really reminds me that while we thought that this was going to be a relatively straightforward endeavor to use ChIP-seq in primary tissues, it took us about 2 years to get there. It had been done quite a bit in cell lines before we tried this, and we thought it was going to be relatively straightforward to port all of that over to tissue, but for various reasons, it wasn't. And to get reproducible data took us about 2 years and a whole team of folks, as you can see on this slide. So again, speaking very generally, this is the type of thing that we started looking for. Really relatively straightforward. We're taking these different states, state A, state B, could be normal tumor, tumor to metastatic disease, and we're just asking the question, are the epigenomic profiles static, as shown here, where we're seeing things that don't change from state to state, or are these profiles dynamic? Are they changing from state to state? And that would be shown here, where you have perhaps binding of an element here that's then lost, or no binding here and then gain. And then we can create a census across all of that and tally all those sites. This is about 6 years of work projected onto one slide, and this is from a paper that we published about 3 or 4 months ago now. And again, this notion of different states in the rows, so normal prostate, localized prostate cancer, metastatic castration-resistant prostate cancer, or I'm going to refer to as CRPC. So the different states and the different marks that we used to annotate those states. So these are transcription factors here, the androgen receptor, FOXA1 and HOXP13, all transcription factors that we know are important in the biology of the disease. And then this mark, K27 acetylation, active promoters and enhancers. And these 4 columns are really what I'm going to be talking about. But one of the ways that we conceptualized of this is that this is sort of like a prostate cancer ENCODE project. And so this is the first story I'm going to tell you. Again, our data is demonstrating that during the metastatic transition, there are fetal developmental programs that are being reactivated. And here's the data behind the story. So one way that we've been conceiving of epigenetics is to think about our own cells and our own body, which all contain obviously the same DNA. We started off from a single cell. And that cell has to differentiate and create the hundreds of different cell types that make up that are comprised in the human body. And that's really enacted through transcription factors turning on and off genes, these master transcription factors. So in any given cell, there's a subset of genes out of the 20,000 or so that's turned on that make these cells have very different functions and look very different. But one thing that I'll come back to is this notion of memory. And as cells are traversing and differentiating, are they recording and keeping track of what programs are being turned on and off as they traverse? So again, it's just a concept to keep in mind as we move forward here. Another way to conceptualize this that I think is really a nice way to think about differentiation that was first proposed by Conrad Waddington, shown over here, is this notion of a marble rolling down a contoured hill. And this is an undifferentiated cell. And as cells differentiate, they roll down the hill and come to land in one of these troughs. There's a couple things that I want to think about as what this is saying. One is that the marble's rolling down. So once you're here, you're downhill and you're stable. Like this appears to be a directional process. And once you're in one of these troughs, you're a differentiated cell. And what this picture is showing is that our cells aren't flipping in and out of their cell identity states. They're pretty well canalized here. And this is something that we started thinking about more and more with our data, about cell identity and how canalized that is and how important that is. And the job of the cell to retain its cell identity is not trivial because it's obviously exposed to a lot of endogenous and exogenous insults and still has to get through that and maintain its cell identity. Now, this slide is really some historical perspective on this notion of development in cancer. And we're not the first people to think about this. This is one of the earliest papers I could find by a Scottish embryologist, John Beard, who really was one of the first, I think, people to publish looking at the relationship, or at least making the connection between development and cancer. He proposed that cancer arose from what's called an irresponsible trophoblast. This is proven not to be true, but I think what's important was that he was one of the first people to publish, again, to tie these concepts together between development and cancer. And I just want to spend a minute talking about this really beautiful paper that came out of Bob Weinberg's lab about 15 years ago. Because as we were going through our data and as our data were coming out, we were trying to contextualize it. This is one of the key papers that I keep going back to that I think really captures an important element of what we're observing. And the experiment here is as follows. They took the exact same genetic elements and put those exact elements into different cell types, and then asked, what's the behavior that I see across these different cell types? And they put these defined genetic elements into melanocytes, mammary epithelial cells, and fibroblasts. And they asked and they injected those into mice. And almost all of the cell types formed tumors. But what was really critical and what they focused on for the rest of the paper is shown here under metastasis. It was only the melanocytic cells that had the capacity to drive metastatic disease. And again, what they concluded from this paper is that the metastatic proclivity of melanoma is attributable to lineage-specific factors expressed in melanocytes and not in other cell types analyzed. And these findings indicate that melanocyte-specific factors present before neoplastic transformation can have a pivotal role in governing melanoma progression. So again, this notion that your cell type, what's embedded in your cell identity, can be predictive of metastatic behavior. And I think that that's really a very elegant experiment and a profound statement. So on to our data. So what I'm showing here is the androgen receptor syndrome and the similarities of the androgen receptor syndrome across these different states. So each dot here is a sample, and the color assigns its state. And the samples that are the closest together are the most related in terms of what their androgen receptor profile looks like. And so hopefully what you can appreciate here in this principal component plot is that normals tend to cluster together, localized tumors cluster together, and metastatic tumors cluster together. And again, it didn't necessarily have to be this way. But one thing that this is saying is that if you take metastatic tumors from different people, their metastatic tumors from the perspective of the androgen receptor syndrome looks more similar to each other than it does to the tumor that it arose from, and that it does to their normal prostate, AR syndrome, within the same individual. Similarly, if you look at active promoters and enhancers, and you do the same type of exercise and ask, through the perspective of active promoters and enhancers, what does it look like in principal component space? Well, we see that, again, localized tumors tend to cluster together, and metastatic tumors tend to cluster together. And what we can do is, as I showed you in the previous one of the previous slides, is that we can take a census and ask how many of these sites and where are those sites that are distinguishing these different states. So that's what this graph shows. There's about 17,500 sites across the epigenome where the AR is moving from localized disease to metastatic disease. So these are 17,500 sites where the AR is sitting in new places in the metastatic state compared to the localized state. And I'm really going to be focusing on this set of sites for the next couple of slides. I'm not going to focus on the sites that are lost. I'm really going to focus on these sites that are gained. And one of the sort of unanticipated things is we can ask, well, what's happening with the other transcription factors that are also, can we chart what's happening to them across these state transitions? And again, we didn't know what to expect, but what this is showing us is that FOXA1 and HOXB13 are not changing nearly as much as androgen receptor. And so one of the things that we can then do is to take those androgen receptor sites, those 17,500 sites that are gained in the metastatic tumor, and we can ask, what do those 17,500 sites look like in normal prostate and localized tumor? So we know that at these sites, almost by definition, or in these states, almost by definition, that androgen receptor is not there because these sites are gained in metastatic tumors. But is there anything there in normal and prostate, or normal prostate and localized tumor? So here what we're looking at is these 17,500 sites. Each row is a site. And we can ask, what do these sites look like in normal prostate and localized tumor through the lens of HOXB13? And one of the things that became quite clear from this analysis is that the sites that the androgen receptor is destined to go to in metastatic tumors is already pre-bound and bookmarked by HOXB13, even in normal prostate, as well as localized tumor. So while AR isn't there, HOXB13 is, and so is FOXA1, and as you might imagine, ATAC-seq. So one of the things that this told us is that where AR is destined to go in metastatic tumors is not random. It's already pre-bookmarked by other factors. The next thing that we wanted to know is, well, yeah, these things are changing, but is it correlated with any other phenotype or any type of function? And how can we look at that? And this was our attempt to do this. On the x-axis, we have differential gene expression between metastatic tumors and primary tumors. And on the right side, these will be genes that are more highly expressed in metastatic tumors compared to primary. On the left side, the reverse. Each dot here is a gene. So all these genes on the right in gray are overexpressed in metastatic tumors. And what we did was that we asked the sites that are gained epigenetically, where do those land? And we projected that onto this map. And hopefully what you can appreciate, and again, here I switched between androgen receptor and K27. There's a huge overlap between the sites that are gained in K27 and AR. So here we just looked at K27 acetylation, and hopefully you can see that most of the sites that are gained in K27 acetylation are overexpressed in metastatic tumors. And this was our first clue, this analysis, when we started to look into development. So you can take the sites that are gained in metastatic tumors, again, these 17,500 or so sites that are gained in metastatic tumors, And you can put them through an analysis. This is an analysis we used called a grade analysis that was developed over at Stanford. And it's essentially, the way that I think about it is a gene set enrichment analysis for non-coding elements. And so when we put those 17,500 sites into this analysis, the terms that come back that are the most enriched and that are highlighted here for both the gained K27 acetylation and the gained AR binding sites are in prostate tumor development. Similarly, you can do a motif analysis and ask just computationally, what motif is enriched across these 17,500 sites over random? And the top motif that emerged is ZEB1, which is a transcription factor, again, operative in EMT, which is important in both development as well as metastatic processes. So this was one of the ways that we showed that epigenetically, these sites really reflect fetal development. And the experiment that we did is as follows. We took the metastatic gained K27 acetylation sites, and then we compared that map to 37 other tissue types that were derived from the epigenome roadmap. So these are a bunch of other tissue types that have been annotated with K27 acetylation. And you can just do this exercise where you ask, where does my set, what set of sites that I've identified, what do they most resemble? And that's shown here in this heat map where each row is a different tissue type, either from fetal tissue shown in red or adult tissue shown in black. And you can ask, for our sites that we saw, how similar are they to each one of these 37 different tissues, with their similarities shown, highest similarity in red, lowest similarity in blue. And hopefully what you can see is that these two replicates here are the most similar to our sites. And this is fetal urogenital sinus. This is the tissue precursor that gives rise to the fetal prostate during development. And then as you sort of walk down this list and look through, the next most similar is small intestine, large intestine. And I needed to go back and dust off my embryology textbook. But what's known is that the prostate actually buds off from the fetal intestine. So maybe it's not so surprising that this also shows up here. And also, interestingly, adult prostate really doesn't look similar at all. And just for the sake of time, I'm going to go through this relatively quickly, this part. This is just now looking at the same data that I showed you in a different way, where we just pulled out the fetal tissue and where we looked at the resemblance between the sites that we identified and how similar they were to these different fetal tissues. And again, you can see the urogenital sinus, the precursor tissue of the fetal prostate, coming out as being the most significantly similar. And then just to make sure that this wasn't some generic metastatic program, we wanted to do this exercise in a different tumor type that was epigenetically profiled. And so this is a data set from Luca Mignani's group in London, where they had epigenetically profiled from K27 acetylation, localized breast tumors and metastatic breast tumors. We took the sites that were gained in metastatic tumors by K27 acetylation and did the same type of analysis that I showed you. The top tissue here that the metastatic breast tumor K27 sites are most similar to placenta. So different ordering than we saw with prostate. So we felt that this was a very prostate specific program that was being reactivated. So I'm going to close this story. This is just an article that came out in the New England Journal last week that describes the work that we did in this paper. And I think that they did a nice job summarizing this, again, across these different states: normal prostate, localized prostate, metastatic, how things are changing, and how similar the metastatic site, metastatic epigenome is to the fetal prostate epigenome. And this really sort of reminds me of Jacot's famous quote here that nature's a tinkerer, not an inventor. So it was not the case that these metastatic tumors are just inventing a new program or opening up closed chromatin or using another set of sites from another cell type. It was rolling sort of back up the hill. So that's at least what our epigenetic data suggests at this stage. So that's the first story. The second story that I want to focus on, I'll probably only spend about 5 or 10 minutes here because a lot of this is already published. We wanted to know too, not only just can we understand the landscape and project these landscapes across these state transitions, but is any of this functional? Does any of this matter from a functional point of view? And one of the first things that we did was that we intersected our epigenetic data with data that was being generated in Matthew Myerson's group, and that was worked on by Srini Viswanathan, who's now a faculty member here. They were looking at the genetics of the transition from localized prostate cancer to metastatic prostate cancer. And what this plot says over here, and I'm not going to get into the particulars or the specifics, what we did was that we intersected where there were copy number alterations here, and we intersected those regions with H3K27 acetylation gain in the metastatic state. So really taking a genetics and epigenetics approach, and the top features that come out of that exercise are shown here in this end of the graph. And the androgen receptor is one of the top, and then there's also a lot, again, these transcription factors that are important in prostate cancer biology. And again, I'm going to walk through this story pretty quickly. We published on this a couple of years ago, but essentially this was one of the things that convinced me that there can actually be non-coding drivers for important genes. And this really is a story about the androgen receptor and an enhancer that we identified as being an important element and a functional element. And it's been known for 25 years that under the selective pressure of androgen deprivation therapy, one of the ways of resistance is to amplify the androgen receptor. And for many years, people had hypothesized that it's the gene that is driving that resistance phenotype. So again, we can go back and look at our data, and we can look at certain locations and look at our differential acetylation sites so that we can identify areas of gained enhancers and promoters as you transition to the metastatic state. And this was one of the regions that we looked at. Here's localized prostate cancer in blue. There's 6 samples, each row is a sample. 4 metastatic samples in orange here, each row is a sample. Here's the androgen receptor. This is a screenshot of about a little more than a megabase. And what you can see is K27 acetylation peak at the AR promoter in both the localized and metastatic states, but about 650 kilobases away, you see much less acetylation here and much more acetylation. So this is one of these gained acetylation sites that I've been talking about as you transition from localized to metastatic disease. This is the genetic data from Srini and Matthew. And this is a copy number plot. So higher copy number denoted on the positive y-axis. This is the physical location on the x-axis. Here's the androgen receptor gene. And here's the area that intersects with that gained acetylation in orange. And what this is meant to show you is that there's this whole region is amplified. And if anything, there may be a little bit more amplification here than here. And when they looked a little bit more closely where the AR was amplified, they found really 2 scenarios. Either the androgen receptor gene and this putative enhancer were co-amplified. That happened the majority of the time. And about 17% of the time, they did not find any amplification of the androgen receptor itself, but they did find amplification of the enhancer. And they showed that in these cases, even when only the enhancer was amplified, that the RNA levels were still very high for AR. And again, I'm not going to get into the specifics of this because it's been in the literature now for a while. but we use this methodology called chromosome conformation capture, which allows you to identify which regions are physically interacting with each other, despite the fact that they may be far apart in linear space. And what we showed was that our candidate acetylated enhancer was strongly interacting with the androgen receptor promoter. And then David Takeda did some very elegant experiments where he looked to see either by Cas9 or CRISPR interference through CRAB, if we suppress this enhancer region, would we see a decrease in androgen receptor levels? And we were able to do a proliferation-based readout screen because we know that the cell line that we're working in is dependent on the androgen receptor. And so if you knock the androgen receptor out, either through its enhancer or through its promoter, that the guides that are carrying that particular perturbation, if it decreases the androgen receptor, that over time, that cell will be selected out of the population. And so if there's an area that's an enhancer that's functional for AR, this would be at time 0. And over time, we would expect the enhancers that are functionally relevant, that are carrying that guide, would be selected out over time. And you can measure that. And in fact, that's what David did, again, for both CRISPR-I as well as CRISPR-Cas. We tiled this region very densely. It's a 9 kilobase region. We tiled about 900 guides across this region. And for each guide, we get a score for how depleted it is. The more depleted, the higher deflected it'll be on the y-axis. So there were 3 regions that were really strongly depleted. Those regions aligned almost identically with the DNase hypersensitivity areas of open chromatin. The acetylation was much broader. The acetylation went across the 9 kilobases. What this screen showed us was that the signal really localized to these areas of accessible chromatin. And again, just for the sake of time, I'm not going to get into this, but David also, through CRISPR technology, put in an additional copy of the enhancer into the LNCAP prostate cancer cell line. And long story short is that overexpressed the androgen receptor gene itself and phenocopied what we know androgen receptor overexpressing cells, how they normally behave. So these cells were more resistant to things like enzalutamide. Stepping back now, though, we're starting to think about how we can put all of these different data types together so that they articulate particular hypotheses that allow us to then functionally interrogate regulatory elements. And this is now the way that we're thinking about this. So I showed you about AR, and in a couple slides, I'm going to show you with HOXB13, another one of these areas that tends to be both genetically gained as well as epigenetically gained in signal. And so one of the things that I think is interesting here is that if you look at the genetics, the area that's encompassed by the genetics, or the level of resolution, is about a megabase. And it contains about 20 genes or so. If you look at now across this area and ask, well, if I'm going to layer on the epigenetics now, and where has acetylation gained in the metastatic state compared to localized tumor and normal? Well, that's shown in this track here in red. And what we can see is that there's a lot of acetylation activity over here. And you can blow up that region. And so we've gone from about a megabase now to about 150 kilobases. And then now we can add on looping data, or this so-called chromosome conformation capture data that I talked about before. And that allows you to take these regions now that we've identified through genetics and acetylation and tether them to a particular gene and provide a very clear-minded hypothesis about what we can go ahead and functionally test. So what the totality of these data are telling us are that this is an area that's genetically amplified. It's also gains in acetylation, and those regions that have gained in acetylation loop to HOXB13. So then we hypothesize that if we perform CRISPR interference or CRISPR-Cas9 against these regions of interest that are supported by these different intersecting data types that would affect HOXP13 expression levels. And indeed, that does happen. So on the x-axis here, we have controls. We have various guides against HOXP13 against these different regions that correlate to these 1, 2, 3, 4, and then HOXP13 mRNA on the y-axis, cell proliferation and growth curves as well. So we're starting to sort of adopt this technique for areas of interest now for trying to interrogate the functional relevance of some of these regulatory elements. And then finally, in the remaining time, just checking the time here. So in the last 10 minutes, I'm going to talk about a story that we also just recently published a couple months ago that really, again, uses epigenetics and uses the feature of epigenetics, really, I think, cell identity, uses these features of the epigenetics of cell identity to detect, to sensitively detect tumors. And again, I come from a genetics background, and I just put this in there because this is my own personal feeling, is that I think we're going to make the biggest impact on cancer-related mortality through things like early detection. I think that's a huge challenge, but this is my own sort of personal belief. It's what motivated me to even get into genetics. And there are folks like Bert Vogelstein is one of my scientific heroes, who's also sort of now articulating and advancing the argument about allocation of funds towards early detection and prevention. And again, I mean, cost of drugs is really expensive. I think it's really important work, but again, just really trying to make this argument about resource allocation and impact on mortality. So we wanted to test out a method and look to see if DNA methylation can be used to identify and classify early stage kidney cancer. And the reason that we used methylation is what I had alluded to before, is the fact that epigenetics in general, and certainly DNA methylation, is cell type specific. So every single different cell type has a different methylation fingerprint and pattern, and different tumor types have different methylation patterns and fingerprints. And so now, as opposed to just using, say, genetics to look, we can start to think about using epigenetics because it may be a little bit more tied to cellular identity and state. And again, just for the sake of time, and because this is published, I'm not going to get into this assay in excruciating detail, but this was an assay developed by Daniel DiCarvalho's group over at University of Toronto. And it's based on immunoprecipitation of methylated DNA. I think that's really the key point here. So there's a lot of assays out there right now in the liquid biopsy space. Most of them are based either on just isolating the DNA and sequencing it brute force. Some of it's based on hybrid capture. This is an assay that's based on immunoprecipitation. I think that those differences are meaningful. Happy to talk about that offline. But for methylation, we're essentially, the signal that we're looking for is represented here. These are samples that don't have cancer in red. Each row, again, is a sample that doesn't have cancer. These 5 rows here, 5 rows in blue, are patients with cancer. And we get a methylation map for each individual. And we're looking for areas like this, areas that are differentially methylated, differentially methylated regions, or DMRs, that are present in one state and not present in the other state. And one can go through that and identify those. Again, this is a volcano plot similar to what you saw before. On the x-axis, each dot here is a methylation site. The x-axis is the degree of differential methylation in cases and controls. Cases greater than controls, controls greater than cases on this side, and the significance of each one of those regions. And you can tally them and draw a line and show which ones are significant. This is from Daniel's paper. He did this for pancreatic cancer, looking at 24 cases versus controls. You can see that there's lots and lots of differentially methylated sites. And you can ask, do those differentially methylated sites classify cases from controls? Indeed, they do. And then you can ask, how well do they actually classify if you take 300 and you try to hold out particular cases or controls and then just use their methylation pattern to classify them. And that's shown in these ROC curves. Again, this is Daniel's data. And the interesting point here is not only can he just detect late stage pancreatic cancer, but also early stage with very good classification and accurate classification. And again, this is a very, very cluttered space right now, driven by different approaches, different analytes that you can look at. And again, I'm just gonna skip over this quickly for the sake of time. We think that there's good reasons to use this particular type of methodology over others, although that's open for further discussion. One of the reasons that we think that it is quite powerful is that you can envision this as thinking about looking for a needle in the haystack. One of the differences that we want to identify and measure between cancer cases and controls. And I think what epigenetics allows us to do in a way is to pull out, there's more needles to look at compared to genetics. And again, I don't think I need to convince many people of this, that the earlier that you detect a particular tumor type, the better their prognosis. About a third of kidney cancers are diagnosed when they're not localized. And their 5-year survival shown over here on this side is much worse the later you detect. And this has been held true for decades for almost any cancer. This is just to show that kidney cancer has a particular challenge in the circulating DNA field in general. So this is an article taken from the experience with Guardant, where they looked at 22,000 cancer cases distributed along the x-axis here. And on the y-axis is the amount of circulating tumor DNA for each different tumor type. And for whatever reasons that we don't understand, kidney cancer tends to be a low shedding tumor. So, and again, I'm just gonna sort of skip over this and just get to the punchline, which is there's many more recurrent epigenetic changes in kidney cancer than there are genetic. And again, I'm not, just because for the sake of time, I'm gonna go through this quickly. I think this type of thing needs to be done more. We did this experiment with David Kwiatkowski and Tony Schwery, where we did the following: take the same exact cell-free DNA, half goes to approach A, half goes to approach B. So as I spoke about before, part of the sample went to look at hybrid capture-based detection. Half went to cell-free methylated DNA. The sensitivity with cell-free methylated DNA was better than with hybrid capture. So in the last few slides, these are the actual data that we generated. So 69 cases with kidney cancer, 13 controls. This is a really, really important point. We need to get more controls. It's early days with this. There's an age difference between the cases and controls. That's important. We need to think about this more and to understand this and age match it better. So that's a limitation. But I do want to note that about a third of these samples were early stage 1 and 2. We can do the same type of exercise that you saw before, have a set of sites that are gained and lost, differentially methylated regions. And take those top 150 and bottom 150. And again, this is open for why 150. There's more work to be done there, but we can ask, do these differentially methylated sites distinguish cases from controls in a principal component plot? They do. And then we can more formally test classifications through these 80%, 20% training test set exercises, which allows you to build these ROC curves. And again, just for the sake of time, I'm going to skip over these details, but I'm happy to talk about any of that offline. And what this shows here, this is the data that we generated. So each column here is a sample. Each sample is given a methylation score, and they're color-coded here by whether or not they're cases in blue or controls in gold. And you can ask, are the methylation scores for the cases different than the controls? By and large, the answer is yes. Yes, you can see all these cases up here, the controls down here. There's about 5 or 6 cases and controls that are more difficult for whatever reasons that we still don't understand to classify. Interestingly, these two cases were the 2 smallest stage 1 tumors. We did not see a correlation of score by stage, but we also don't have a lot of samples, so it's hard to make any definitive statements. But again, very high classification accuracy here, ROC 0.99. And then finally, this is the last slide or two. We also wanted to say, well, can we not only distinguish kidney cancer from healthy controls, but can we distinguish kidney cancers from another GU cancer? So we looked at bladder cancer. This is a little bit better age matched. These happen to all be metastatic bladder cancers, but same type of thing. This methylated DNA signatures are able to distinguish and classify kidney cancer from bladder cancer, again, with a very high ROC. And I think it is exactly, well, at least 12:56. I tried to acknowledge people as I went through, but I'm sure I didn't do justice. So this slide really captures all of the people that have been critical to performing the work that I just presented. So I'll stop there. And with that, I'm happy to take any questions.
Hey, Matt, thank you for a spectacular talk. I'm glad the computer held up. I was worried there a little bit in the beginning. So there are a couple of questions. One related to the HOXB13 and FOXA1 still bookmarking, these sites that become open later, even though AR is not binding to them early, but it's binding late. Are there any changes in FOXA1, HOXB13? Is it possibly just somewhat of an increase in them that is, tipping you over, or is it not related to that?
Yeah, so there are 2 things that I'll say. One is that at this given level of stringency, to look at peak differentials, AR is disproportionately different than FOXA1 and HOXB13. Now, FOXA1 and HOXB13 do move around, but nowhere near. And as you relax the threshold, you can see sites, but it's probably at least an order of magnitude less. They're moving around much less. What I will say, in data that I didn't present, and there's a paper on bioRxiv now that was really driven by Sylvan Baca, a postdoc in the lab, is that when you transition to neuroendocrine prostate cancer, and in this way, we're viewing it as under really, really severe selection pressure, undergoing lineage plasticity to the neuroendocrine phenotype, which is a huge, huge transition. There you see huge shifts in FOXA1. And there I can tell you, we see probably about 35 to 40,000 sites that are shifted, about 25,000 sites, I think, that are gained in the neuroendocrine state for FOXA1, and about 15,000 sites that are lost.
So just as a little bit of a follow-up to that, so what in the metastatic setting, or I should say in the primary setting where FOXA1 and HOXB13 are there, I think you said that chromatin's not open though. By ATAC-seq, it tends not to be open. There's no H3K27 acetylation. Do you have any data on DNA methylation or H3K27 trimethylation, or other things that might be actively keeping the site closed?
Yeah. So great question. I misspoke that there is ATAC. It is open. It's accessible, at least as measured by ATAC. in normal prostate.
Okay.
And whether or not that may be a little bit more open in the metastatic state, I don't know. But to your point about methylation, these sites are absolutely hypomethylated.
Okay. You didn't misspeak. I think I misheard. I think you probably did not misspeak. A second question, kind of technical, that in these high content tools you're using, you kind of assign a site to a gene based on proximity. And, I mean, to what extent do you think that is really valid, or how much of a problem is that?
Yeah, great question. I think that this is obviously a key question just in the transcriptional biology field, period, which is how do you start connecting regulatory elements with their target genes? So, I've gone back and forth with this. There's a lot of techniques now to sort of do this. And I think where I stand now is that these different techniques, such as distance or chromosome conformation capture, or even genetics, so like expression quantitative trait loci, where you can sort of look at the correlation between genotype and gene expression levels. I think those are all ways to connect regulatory elements with target genes, as well as CRISPR screens, right, where you can start thinking about perturbing these elements and seeing what genes they affect. So I think all of those are useful. I think they may be useful, differentially useful, in different scenarios. And just to put a little bit of a fine point on that, for example, I would never use 3C-based types of technologies to look at something within a window of, like, say, 10 or 15 kilobases. I would, use another technology. So again, I think it is a really important point, and we can't assume that distance is going to really, at the end of the day, tell us for sure what gene, but it's also not completely not informative. So I think that we'll start, to me, I think the strongest thing would be perturbation experiments followed by careful measurements of gene expression.
So one other quick question that just popped up. So if circulating tumor DNA is from dead apoptotic cells, which presumably it is, are the differentially methylated regions in these dead cells representative of the actively dividing cells in a tumor?
Yeah, that is a great question. What I can say is, yes, a lot of the background is due to white blood cells. And so we're also looking at ways now, and again, I actually do want to say, I meant to say this at the beginning, that if anybody's interested in any of this work and wants to think about things further, we would love to hear from you. There's a lot of data, and I think a lot of different directions. So the more people that and eyes on this, or projects or collaborations, the better. And the person that asked this question, this is now we're trying to be a little bit smarter about the way that we analyze this data, because we do realize that there is a background. And can we start using these patterns that we know exist in a more tissue specific way, or can we use data from the tissue itself and from the white blood cells to sort of enhance our signal to noise ratio, to decrease the background and really look at what the signal that we want to look at. So we're looking at ways to do that. What I will say too is that when we look at the correlation between the differentially methylated sites that we get out of these types of exercises, and ask how similar are those to, say, kidney cancer TCGA methylation, there is a significant correlation between what we see there.
Okay, and we have 1 minute left. I have one more big picture question. Just going back to the developmental reprogramming. Any speculation on why prostate cancer at metastatic sites or after castration resistance needs to go back to that early developmental stage versus primary prostate cancers, which maybe they don't need to do that?
Yeah, thank you very much for saying that, because I meant to also say that. And this I didn't say? Because, and again, this is just a hand-waving hypothesis. So, but what I do think is that there's a lot of similar traits that need to be tumor to become metastatic. Some of those traits, I think, are really similar to what goes on during embryonic and fetal development. It's a time of tremendous movement and invasion. So these are traits that I think become active during development, and then they become quiescent. And then when tumors, become invasive and they need to move around a lot, and again, this is just a hypothesis, they end up reactivating those programs. So you have this sort of like on, off, but still memory, intact memory, and then back on again. It's somehow, and what that switch is between off and on, again, if anybody has any ideas about how to think about that or mechanistically explore that, we would love to hear from you.
Okay, we've hit the 1:05 mark. So if you've heard Matt asking for, inviting you to send him questions, comments, emails, potential for collaboration and interactions. And Matt, thank you very much. You've, as always, given us a lot to think about. Great talk.
Great.
I enjoyed it very much.
Thank you so much. Really appreciate it.